All right, hi everyone. Thank you very much for joining us today. What we want to talk to you about is some of the work that we've been doing to preserve species affected by myrtle rust on the east coast of Australia. And hopefully you'll find something in the presentation that you can use for preserving species in your own area. So Veronica and I both work for the Botanic Gardens of Sydney, and that's made up of 3 botanic gardens, one in the Blue Mountains Botanic Garden at Mount Tomah on Darug country, the Royal Botanic Garden Sydney on the harbour on Gadigal country, and the Australian Botanic Garden at Mount Annan on Dharawal country. So I'd just like to start by acknowledging the traditional custodians of those lands and paying my respects to all the elders. So at the Australian Botanic Garden at Mount Annan, we have the Australian PlantBank, which is a conservation and research facility, and that's aimed at preserving our Australian native species. The heart of the seed bank has, the heart of PlantBank is a seed bank, which has collections of seed collections for over 5,000 different Australian native species. And we also have facilities for tissue culture, cryopreservation, and access to facilities for growing live plants. So the purpose of PlantBank is to hold collections of species to guard against extinction, and also to provide material and the knowledge that's needed to return species to the wild if they happen to disappear from their local populations. And one of the groups, of course, that we're having to do a lot of work to preserve at the moment is the Myrtaceae. And our major threat is myrtle rust. Now, I'm sure you've seen something like this before, but just to recap, myrtle rust was first detected in New South Wales in Australia in 2010, and within 5 years it had spread all the way to the top of the country and all the way down to the bottom. And this is a huge problem for Australia because the Myrtaceae is one of our dominant plant families. We have around 1,700 species and many more subtaxa, and more than 382 of those species have been found to be susceptible to the rust. So it's a huge problem for us. And it's becoming a bigger problem because we're finding that some species are particularly susceptible and are on their way to extinction already. And this paper in 2021 indicated that there were 16 species that are likely to disappear within a generation. So we really need to use ex situ conservation to support the preservation of those species and to provide at least some material that we can use to maybe return those species to the wild when we've sorted out problems with myrtle rust. So seed banking is a really efficient way to conserve many of our Myrtaceae species. It's effective for what we call orthodox seeds, and those are seeds that will tolerate drying down to around 3 to 7% moisture content, and then they'll tolerate storage at -20 degrees Celsius. So these species are common in dry environments, and they're common for dry dehiscent fruits. So some of the genera that are suitable for seed banking in Australia include Angophora, Beckia, Callistemon, the eucalyptus species, Leptospermum, Melaleuca, all those species that grow in dry environments and that produce the dry dehiscent fruits. Myrtaceae in wet environments are a little bit more complex, and so we have a mix of species that have orthodox seeds, so Lophostemon, Melaleuca, Syncarpia, for example, and then we have species that produce what we call recalcitrant seeds. So these are seeds that are completely intolerant of drying and we can't bank them at all, and that includes Eugenia and most species of Syzygium. And then we have this other mixed bag that we call intermediate, and that includes species that will tolerate partial drying, or they'll tolerate complete drying but don't like being stuck in the freezer, or they don't live for very long when we do put them into storage in under standard conditions. And that includes a lot of the species from our rainforest that are particularly endangered by myrtle rust, and that's Rhodomyrtus, Rhodamnia, Rhodomyrtus, and some of the other Lenwebia, Rhodomyrtus, and other species. So we discovered a little bit by accident that some of these species didn't like being stored under standard conditions. So we had these species, we tested them after drying, they were completely tolerant of drying, we put them into storage and then pulled them out of storage within a few years and discovered that the germination had declined significantly. So where these examples here on the screen that you can see in front of you, on the left you can see what the percentage germination was after drying, and on the right the percentage germination after freezing. And here's an example of what's happening here with Rhodomyrtus psidioides. So seeds that we stored at 15 degrees Celsius for a few months were fine, but the seeds that we stored at -20 degrees were completely non-viable at the end of that period. So this was a bit of a surprise, and a very, very unwelcome surprise. And what we wanted to do from here was to find out what's going on. Why are these seeds so short-lived in storage when they're so tolerant of drying? So we started a project to look at what might be happening during freezing and thawing that was causing that loss of viability. So what we've been using is a machine called a differential scanning calorimeter, very, very fancy name for a machine that will basically lower a seed sample from room temperature down to -150 degrees, and then back up to 50 degrees Celsius. And what we're doing there is recording any energy changes that are happening as the seed's freezing and thawing. And the output we get is a chart that shows you those peaks and the temperatures that they're happening at as the seeds are freezing and thawing. And this project started out as a collaboration with Christina Walters at the National Laboratory for Genetic Resources Preservation in Fort Collins in Colorado. And then as it progressed, we got funding from the Ian Potter Foundation to continue on the work. So here's an example of the sort of output that you can get. This is a species that we know is short-lived. The line down the bottom is showing what's happening as the seed is cooling, and in this case, not very much. And then the line on the top is showing what's happening as the seed is warming. Here is the standard storage temperature at -20 degrees. And here you can see this great big melting transition that's happening at the temperature that we usually use for standard storage. An example of a long-lived species, this is a Syncarpia. And again, we're just showing the warming curve here. Here's -20 where we store the seeds. And oops, let me just go back there. Uh-oh. Sorry about this. All right, just going to click through. I obviously clicked the wrong button there. Okay, so here we are, back with Syncarpia, and nothing much is happening at -20 degrees. And this species is still germinating really well after 15 years in storage. Okay, so when we look at 4 of the species that we've been having difficulty with in storage, if you look at these charts, the red line is indicating -20 degrees, our standard storage temperature. And you can see just to the right of that line, this huge melting reaction that's occurring as the seed is warming. So where we were thought that we were storing these species at a temperature where they were going to be stable, we're actually storing them in this state where they're very unstable. And this is caused by the reaction of lipids in the seed. Seeds can have a lot of storage lipids that help them with germination and growth. And these are the things that are reacting at those temperatures. Where you can see the blue shaded bar over on the left, you can see there's not much happening below -100 degrees. And for 3 of the species, we also had not much happening at around -5 to 4 degrees. So we thought possibly that these might be alternate temperatures that we could try for storing the seeds to see if we get a better response. So these are some examples of seeds that we've tried storing at alternate temperatures. This one is Rhodomyrtus beckleri. And our first experiments on this, we dried the seed to 3 different moisture contents, to 3, 5 and 8%. There was no significant difference between germination after drying for any of those, but there was a difference in the response to storage. So this particular one was stored at 8% moisture content for 4 months. And you can see here that it's already lost viability and when it was stored at -20 degrees. And the best response we had was in storage at -5 degrees. So for this species, it did well in storage for 7 years at 4 degrees. Also looks like it will do well at -5 degrees. So it looks like the optimum for that one. For Lenwebia species main range, we had no significant differences among the treatments that we've tried so far. That was just after storage for 4 months. But there was a slight decline in healthy shoot production with decreasing storage temperature. So we've got another experiment where the seeds have been stored for 12 months. We're just wrapping that up at the moment, and we'll be able to see whether longer term storage will produce more of a difference between the temperatures. Rhodamnia merdeniana, stored for 12 months, there was a clear decline in viability for species stored at the standard -20 degrees Celsius, but we had really good germination after storage at -5 degrees. So that looks like the optimum temperature for this species, or at least the best temperature of the ones that we've tried so far. Rhodamnia rubescens, stored for 4 months, we found better survival at -20 and -192 degrees than for Rhodamnia merdeniana. No real difference between the treatments, but this was in relatively short term, and again we're running an experiment now where the seeds have been stored for 12 months, so we'll be able to get a better look to see what happens in that longer time period at -20 degrees. And I'm expecting germination to decline at -20 and to remain the same for seeds stored at -5. And then our final species was Syzygium anisatum. This one was found to be short-lived by our seed bank manager, Graham Errington, who did some work on artificial ageing. And for this species, we so far have only tested storage for 1 month at a range at -20 and -192, and this species has done really well at those temperatures. So if it does decline quickly at -20, then we have cryopreservation at -192 as an option. Okay, so seed banking is possible for these species. We just need to get the moisture content and the temperature right. The main limitation on seed banking is the seed availability. And what we found is that most of the species that I've been showing you here today are producing very few seeds in the wild, so we just don't have the seed available to bank. And the only way that we can get that seed is to have ex situ collections. So potted collections are needed to generate the seed for banking, to do the research to make sure we've got the right temperatures, and also to preserve species with recalcitrant seeds, and to provide material for preservation in vitro, so for tissue culture and cryopreservation. So I'm going to hand over now to Veronica Vila. I'll just first of all thank the Ian Potter Foundation and the sponsors of the Rainforest Seed Conservation Project for this part of the project, and those people who've helped me in the laboratory and the nursery. And now I'll hand over to Veronica to talk about the ex situ living collections that she's developed for 2 of our difficult species.
Thanks, Carol. Hi everyone, I'm Veronica and I'm a horticultural research assistant here in the Australian PlantBank. Excuse me. I'd also like to acknowledge the Dharawal people, the traditional custodians of the land I work on, and pay my respects to elders past, present and emerging. And I'd also like to acknowledge other First Nations people that are with us today. My talk will focus on 2 important ex situ conservation collections, as Karen said, held at ABG, Rhodamnia rubescens or scrub turpentine and Rhodomyrtus psidioides or native guava. The collections were established in collaboration with Saving Our Species as part of their emergency response project. The aim of the project was to collect germplasm before it was lost in the wild due to their extreme susceptibility to Myrtle Rust. Excuse me. Genetic testing by the Research Centre for Ecosystem Resilience has determined that both collections are representative of the diversity in the wild, which is important for long-term conservation. Rhodomyrtus psidioides once occurred from Broken Bay, approximately 90 kilometres north of Sydney, to Maryborough in Queensland. It was typically found in coastal to subcoastal areas. However, it has also occurred up to 120 kilometres inland in the Hunter and Clarence River catchments and along the border ranges in New South Wales. There is no documented evidence of resistance to Myrtle Rust for this species, and it is expected to be extinct in the wild in 1 generation. Rhodamnia rubescens typically occurred in coastal districts north from Bateman's Bay in New South Wales to inland of Bundaberg in Queensland. It was found in littoral, warm temperate, subtropical rainforest and wet sclerophyll forest. Initial testing has shown some resistance for this species, and work is underway to explore this further. Once again, it is expected to be extinct in one generation, only 13 years after the arrival of Myrtle Rust to Australia. So now to the good news. Rhodomyrtus proved to be fairly straightforward in terms of timing for and ease of propagation. Rhodamnia took us a little bit longer to pinpoint the timing for propagation, which turned out to be winter. Fortunately, both species have a propensity to sucker, so where a parent had no suitable material for cuttings, excavated suckers provided another option, as you can see in that middle image. Suckering also got us thinking about the possibility of root cuttings where there was no above ground material available, and some early work with nursery plants looks promising. That's a root cutting on the right image there. All material was collected and then sent to the gardens via express post. A large proportion of the material for both species came in with visible pustules or lesions, but all material was treated as infected regardless of visible symptoms. Initially, propagation was carried out in the lab in a fume hood to reduce the risk to the nursery collections. New wild collections are still processed in the lab, although it is possible to safely process cuttings in a nursery environment. Both cuttings and suckers were treated with a curative fungicide before moving into the propagation glasshouse or broader nursery, and then monitored closely for a period of about 4 weeks. Generally, no follow-up treatment was necessary. The collections currently represent 95 genets across the 2 species and approximately 800 plants in total, although that figure is dynamic depending on the time of year. Both species do well in cultivation, and repropagation is necessary roughly every 18 months. This image shows the first generation in the background and the first repropagation done last year in the foreground. Besides Myrtle Rust, both species are also susceptible to pests such as mites, thrips and scale, to name a few. I think it's important to say at this point that Myrtle Rust should be viewed as another disease to be managed in the nursery environment. Educating ourselves on what to look for, how to respond, and being prepared in advance can greatly reduce the risks and the anxiety. I'm often asked whether we've noticed any signs of resistance in the collections, but the purpose of the core collection is to secure the material, so we would never allow an infection to continue untreated. The collections provide the material needed to produce cutting-grown plants, seed and seedlings for further research. I can't overstate the importance of record keeping for ex situ collections, from arrival through propagation and then throughout the life of the plant. With such a large collection of plants from multiple provenances, keeping accurate, up-to-date records and labelling is essential. We attach a durable engraved tag as soon as possible after first potting of a struck cutting. Each collection is assigned a unique identifying number linked to our database, and the collection details, propagation information, and any other important information is recorded. Myrtle Rust is managed using a combination of regular monitoring every couple of days, cultural practices, and fungicide use when appropriate. Regular monitoring ensures that when Myrtle Rust arrives, the situation doesn't get out of hand. Cultural methods such as adequate spacing for airflow, pruning to control the size of the plants, considering the type of irrigation, and good plant and equipment hygiene all reduce the risk of infection and therefore reliance on chemicals. Having said that, sometimes spraying is unavoidable. The past 3 years have been exceptionally wet and humid due to La Nina, and there were times when regular spraying was necessary. Having the right personal protective equipment, fungicides, and minor use permits on hand ensures that spraying runs smoothly. It's also important to monitor weather conditions generally and be familiar with microclimatic conditions at your particular site. Fungicides are applied on a rotating basis to prevent the disease from developing resistance to treatment. An understanding of the modes of action for each chemical is essential, as well as training in application. We were delighted to discover that 12-month-old cutting-grown plants will flower and fruit, and in fact, potted plants are incredibly productive. To date, around 60,000 seed from 39 maternal collections have been banked for the 2 species. Prior to these collections, we were unable to establish wild collections of either species due to the impacts of myrtle rust. And there's a really lovely picture of a Rhodamnia in the middle there that was propped last winter, and it's already in bud. So I'm very excited about that. As well as seed for Karen's work and banking, the collections have provided material for numerous research, including metabolomics, cryostorage trials, myrtle rust assays, and the establishment of tissue culture collections. Maximising the use of plant material ensures more bang for your conservation buck. For example, usual seed banking procedure is to run a germination trial with a sample of an incoming collection. Excuse me. Rather than discarding the seedlings that were produced, they were grown on and used for myrtle rust susceptibility testing. In the case of Rhodamnia, some of the seedlings demonstrated the same resistance the parent material had displayed in earlier testing, which was very exciting. Herbarium vouchers capturing flowering and fruiting have also been made from the collection, ensuring that information is captured where the wild plants may no longer exist. Holding the entire collection at one institution was identified as a risk, and work is underway to disperse clones to several other botanic and regional gardens. Dispersal also enables other institutions to conduct their own research with a ready supply of plant material and provides the opportunity to observe how the plants perform in different environments. We believe these collections are a case study for other myrtle rust susceptible species, and work is underway for a similar collection of Rhodamnia maidniana, which is similarly threatened. Planning is also in progress to explore resistance breeding for Rhodamnia rubescens with a view to eventually returning plants to the wild. Thank you so much for listening.
Thank you, Karen and Veronica. That was really, really interesting and definitely gives us lots of hope for the future. So audience, I'm happy to field questions. If you have any questions, just put them in the chat and then and then I can read them out to our speakers. I'll start. I'm just kind of curious about how many like how fast is the process of screening to make sure that these seeds have survived the freezing process? And yeah, I mean, you know, and just because we have, I mean, especially, well, Australia has a lot more of the Myrtaceae than we do in New Zealand. So yeah, it will be great to hear about that.
Yeah, sure. So the initial screening is fairly quick. So we'll test the seed when it's fresh, then we'll test it again after drying to make sure it'll tolerate drying. That will only take a few weeks for the seed to dry before you retest it. And then we'll put a subsample in the freezer and pull it out after 3 months, retest it then, and then again after 12 months now, because we've learned that just because it tolerates freezing for 3 months doesn't mean it's going to survive any longer than that. So you're looking at about 12 months to 18 months to get a really good handle on how the seed's going to respond in storage. But the good news is that in the meantime, you can store, it seems like you can store most species in the fridge at 4 degrees until you figure out how it's likely to respond to storage in the freezer.
Yeah, no, that's really interesting. Also just, I mean, it was also really interesting to see that, you know, that you have such a variation in terms of the temperature that they store, because, you know, I would think like most seed banks just store it at much colder temperatures. Yep.
Well, yeah, exactly. And that was why it was such a surprise to us, because we've worked so long with orthodox seeds that do store well at -20, which is the standard seed banking temperature all around the world. It was a bit of a shock to us to see how poorly these ones had done in such a short period of time. And so we aren't really set up for storage at -5 degrees, so those experiments were done with a little camp freezer. Yeah, we're looking at investing in a freezer for that temperature for those specific species that won't tolerate -20.
So is it, I guess in terms of like of infrastructure, of like making sure that you have enough space to store the seeds, like how if for a -5 freezer, yeah, I guess like how many seeds would you be able to store at once?
I guess that depends on the size of the seed. So most of those species, Myrtaceae species that produce seeds that will tolerate drying, the seeds are fairly small, and so you can store thousands in just a small packet. It doesn't really take up much space at all if you've got those smaller seeds. Things like Syzygium hodgkinsonii, which has a seed the size of a golf ball, well, they can't be stored anyway, but if you could store them, then you wouldn't get as much in the same amount of space.
Yeah. Oh, that's really interesting. So one of the questions that has come in is, you have so many Myrtaceae species, how do you prioritize them?
Basically, we've been prioritising by the level of risk. A lot of the Myrtaceae that we have in the seed bank was banked before Myrtle Rust became such a huge problem. So we have ongoing programs to collect Myrtaceae, and so do other seed banks around the country. But for the detailed work onto storage, we're saving that for the really endangered species, and for the ex situ collections that we're developing in the potted collections in the nursery, because that takes so much work, then that's only for the critically endangered species.
Interesting. So a question is, did you measure the lipid content in seeds susceptible to drying and their tolerance?
Yes, and there'll be a paper coming out soon. I'm just working on it now. We measured the lipid content, extracted the lipids, and looked at the fatty acid content, and we also ran the lipids through the differential scanning calorimeter to see if the profile of the lipid matched the dry seed, and it did for these species. So yeah, there'll be some work coming out published on that in the next few months, hopefully.
Okay, so another question that's just come in. Can you explain again, how do you know a seed would not be storable at -20?
That is purely for, sorry, words escape me. That's purely from experimental testing. So we've tested the seeds when they've dried, we've put them into storage, pulled them out and retested them, and then tested them at a later date, and that's how we know.
Cool. And also it's really interesting about the suckering that you're able to grow the plants for that. Do you, I guess how many of the species do that, or is it fairly common within Myrtaceae to have suckers?
That's a good question. I don't actually know the answer to that. Certainly for these two, it's a real advantage. And I think that might also help or play into the root cutting idea. So even where we have mature plants in the wild that appear to be dead, it may be possible to take root cuttings and then grow suckers from those root cuttings. But yeah, I'm sorry, I can't actually answer the question about Myrtaceae more generally. I'm not sure how many sucker. Hopefully a lot.
Yeah, exactly. I was going to say, I was like, PhD students, listen up. This is a nice project to maybe get involved in. So, it is a question or a comment. It's great to see the plants regenerated using cuttings. Produce seeds. How different or similar are they to their mother plants genetically, especially if we're seeking genetically diverse materials for restoration?
Well, they're clonal, so the cuttings, because the material is taken from the parent as a cutting, it's a clone of the parent.
But the seed material, we haven't done any targeted breeding, so the seed material is just a mixture of the parent, the mother plant, and whatever else it's happened to pollinate naturally in the nursery.
Yeah,
we haven't done any targeted work on that yet, but we do have the collections that we have for those two species are representative of the genetic diversity in the wild. We've had that both the wild populations and the potted collections tested genetically, so we know that's the case. So we've got the potential for targeted crossing there, and to know what both the mother and the father parents are.
Definitely something very important to do. Yeah. Have you found any evidence for genetic variation in susceptibility to Myrtle Rust in screening Rhodamnia psidioides, and have you studied all the genotypes in your collection?
We haven't actually done any of that work.
Mm.
That's kind of outside my remit anyway. So screening has been done by the Research Centre for Ecosystem Resilience in collaboration with PBI.
Plant Breeding Institute at Cobbity.
Yeah, and there's a paper on that. Sorry, what was the other part of the question?
Just whether you've studied all the genotypes in the collection, so they would all be screened at in partnership then?
Yeah, so there's only been a proportion so far that have been screened for Myrtle Rust resistance. So how many individuals would it have been?
Initially there was probably a dozen, and so they were clones, they were cutting grown plants. And then the second lot were seedlings, some of which were from those original parents. So what we saw there was that where the parents had displayed resistance, we also saw that coming through in the seedlings as well. But yeah, we still, ideally it would be great to test the whole collection, but yeah, there's no plans to do that at this point.
Yeah.
We haven't explored the genetic basis for that resistance yet either, but I imagine that's on its way.
Yep, exactly. So a question New Zealand specific. So we have a single Syzygium species in New Zealand, and it has been recalcitrant to seed preservation and highly susceptible. How would you suggest going about preserving the species outside of the field?
Well, I mean, I think probably the first thing to do would be to look at the genetics of the populations, and then closely follow that with some sort of collecting program, so that you can capture that material as quickly as possible.
Yeah, and so establishing a potted collection would be, I think, a key thing to do initially. And then what we're doing with some of our other recalcitrant species or species that we've been having trouble seed banking is we're establishing them into tissue culture and doing some work on cryopreservation. So that's sort of a long-term view, but if you can get them into the potted collections first, then at least you've got the material to work with before the species disappears from the wild and you can't collect it at all.
No, that's definitely good news.
Yeah, and understanding the genetics of what you actually have there, because what we found is when that genetic work was done, we actually found where we had some collections that were actually the same. So it was the same plants. So that allowed us to sort of rationalize the collection a bit. So it's very important to understand the genetics, and it can actually save you a lot of work.
Oh, that's good advice for people thinking about seed banking and preserving our Myrtaceae. Thank you so much for taking the time to give us an amazing presentation. And we'll have a recording available on the Beyond Myrtle Rust website. And it'll also be emailed to everyone who has registered. So our next webinar is actually on September 20th, and we'll be hearing from our PhD student Sarah Sale about her progress in growing rust fungi on artificial substrates. So keep a lookout for the webinar invite in the next couple of weeks. And thanks everyone. Hi, Rara. See you next time.